It's already time. So thanks for our sponsor, Epicipher. Please welcome Epicipher. Yay!
Hi everyone, my name is Cheryl Zaidi. I'm from Epicipher, I'm a project manager for our services group. Like some of you, I help organize and collaborate with researchers interested in running genomic assays to advance their studies. In this talk, I'll discuss how cut and run offers distinct advantages over chip. I'll highlight the importance of key controls to ensure reproducibility and consistency across experiments. And finally, I'll introduce auto cut and run, which enables scalable chromatin profiling, making high throughput research and drug discovery more accessible. So all of these are critical considerations for core labs looking to improve efficiency, reliability, and scalability in epigenetic profiling. So a little bit of background. Chromatin consists of DNA wrapped around histone proteins, forming nucleosomes. Various modifications to chromatin play a crucial role in regulating gene expression. When these processes become dysregulated, they can drive disease, making chromatin regulators valuable drug targets and biomarkers, particularly in oncology and immunology. So there's different types of chromatin regulators, such as histone post-translational modification on the histone tail, chromatin remodelers, proteins that reposition nucleosomes, or transcription factors and other chromatin-associated proteins that bind to the DNA. So we use genomic assays to kind of probe changes in these chromatin regulators. One such assay is ChIP-seq, which is widely used but comes with significant limitation. You're starting with extensively cross-linking over 1 million cells. You're fragmenting your chromatin, which is labor-intensive and prone to variability. You add your antibody, immunoprecipitate your target, but non-specific binding often necessitates harsh washing steps to minimize your background noise. Then you go into library prep and then deep sequencing at 30 million reads to distinguish your signal from noise, but the data quality is often poor. Another limitation of this assay is that it's difficult to automate, resulting in it being relatively low throughput. An emerging and more efficient technology for researchers, especially those in core labs, is cut and run. You have an intact cell or nuclei immobilized to a magnetic solid support. You add your antibody, it diffuses into the cell to find its target. Here we're showing is a PTM. So this is a more physiology relevant environment. You add your PGA aminase. When activated, clips the DNA. This nucleosome complex diffuses out into the solution, so you're separating your signal from noise. You then proceed to library prep and then into sequencing. So with this workflow, you're able to start with fewer number of cells. This is great for those who are working with limited or precious samples. You're sequencing at 3 to 5 million reads instead of 30 million, which can save you on sequencing costs. You also get a better data quality compared to chip. Additionally, with this workflow, it enables you for high throughput experiments for those managing multiple projects with varying complexity. So one thing that's important to Epicipher researchers and those who operate core labs are controls and the standardization of our experiments. So for our reactions, we add 2 spike-ins to our experiment, an E. coli DNA to control for library prep and normalized sequencing data, as well as our SNAP katana spike-ins, which are nucleosomes with bar-coded DNA, which are then conjugated to a magnetic strip adenine bead to mimic a cell on a magnetic solid support. So when these SNAP spike-ins are added into the reaction, they are also pulled down alongside your target, and then the barcodes are also used for analysis. For our cut and run experiments, we also use 3 control antibodies, so IgG as our background control, H3K4 tri, H3K27 tri as positive controls. So they work well with our SNAP spike-in nucleosomes during data analysis to generate these specificity heat map, as shown here, to determine antibody specificity and experimental success. Additionally, you're able to compare these data across different experiments to assess for technical consistency. So how would you implement this assay in your lab? So it's a low-effort adoption with our kits. We offer customers 24 and 48 reaction cut and run kits, as well as 48 reaction library kits that work seamlessly well together using the 8-strip tubes. So in total, these kits allow you to go from cells or samples to libraries and sequencing data in 3 to 4 days. For throughput, most users can comfortably run 24 reactions at a time. We found experienced users can perform 48 reactions with no problem. But what if you're interested in more than 48 reactions? At EpiCypher, we've developed automated cut and run for those high throughput experiments using 96 well plates, making large-scale chromatin profiling more accessible than ever. So here are just some example projects you can tackle with auto cut and run. If you're looking for direct chromatin response to treatment, biomarker discovery in patient samples, or epigenetic signatures of cell identity, where you're mapping chromatin landscapes across different cell types. So in the upcoming slides, I'm going to talk more about the last application that I mentioned. So we developed AutoCut and Run for the Amgen Consortium, a collaborative network of labs across the US and internationally. The scope of this project was to create an epigenomic map of mouse immune cells, with each of their lab focusing on specific subpopulations. There were many samples to process. Not only that, they also had a limited number of cells, which then limited our sample input to 10,000 cells per reaction to cover all 8 of their targets. So samples were collected at Amgen, individual Amgen labs organized at a central lab, and then sent to EpiCypher for auto cut and run and library prep. So with such low input, we had to devise a way, additional way to control the experiment, since we were unable to assess the sample quality beforehand. So to ensure consistency, we included an NIH 3T3 control cell line on each plate alongside Amgen samples. This allowed us to confirm that the experiment was conducted correctly and to monitor assay variability. So this project spanned 2 years, of which we ran 18 plates. That's over 1,700 reactions by 4 scientists. On the right here is our specificity heat map displaying our assay performance, which is averaged across 18 plates, where the max drift between the plates was 6%. At the bottom here, we show the genomic data for H3K4 tri across all the 18 plates, again, demonstrating the reproducibility of the data. And then here is just a flavor of the data revealing unique regulatory signatures in primary mouse immune cells, showing IgG, which is the top, and then H3K4 tri, a mark of active promoters, and H3K27 tri, a mark of repressed genes, for granulocytes, lymphoid cells, and natural killer cells. As you can see, each of these cells exhibit different landscapes. And so if you're interested in bringing cut and run in-house, we have our kits, both cut and run and library prep, validated antibodies for cut and run on our website, as well as our technical support center. If you're interested in high throughput cut and run, we offer services at EpiCypher, where we will run your reactions for you. Or we also, if you're already running cut and run in-house, we have technical webinars for core labs, where we'll go over tips and tricks on how to run the assay. And last, we can help you bring AutoCut Run in-house as a tech transfer as well. If you have any questions, my name is Cheryl from EpiCypher. We're at booth 103. Thank you.
Thank you, Cheryl. Thanks for coming to our session. So our session title is Staying Ahead, How Core Facility Adapt and Connect to Meet Diverse Genomics Need. So we really care about our core efficiency and then how we provide our customer better service, with the newer technology. So we're gonna talk about this here because we are all experts here. So I try to say we can give you the solution to do this, but we don't have it because every core has its own circumstances and then own challenges. So this is more about we share our experience, and then we just discuss our, how we tackle, each challenges together. So how can I do this? Oops. Oh, this is it. Okay. So we have 3 guests, including me today. Eliza is going to talking about the sample preparation at the biorepository, which is really important for any sequencing genomics data. And then Olivia here, she's learning really one of the biggest institute, Genomics Core at University of Michigan. So we can learn from her how she learning this big Genomics Core. And I'm from Cleveland Clinic, and we have very small one. So you can hear some kind of perspective from me learning like small Genomics Core. So I think, and I hope, all 3 talks are very beneficial for you. Okay, first it's Eliza.
Good afternoon, everyone. So my talk is entitled Challenges in Keeping Up with Emerging Sequencing Technologies, but from a specimen collection and a biobanking perspective. So as we all know, technologies are changing faster than ever, forcing us to continuously change and adapt with them. And as we're here to talk about today, troubleshooting how to manage these changes is critical for ensuring the sustainability of the departments and the programs that sponsor these resources. But what I think has been underappreciated in this dialogue is the specimen collection and the biobanking efforts that happen upstream that are often the very source of the samples that are used in these technologies. And as these new technologies come about, they bring with them new chemistries and often new requirements for our input specimens. And so from a biobanking perspective, this presents a particular challenge as the whole goal of a biorepository is to collect tissues in anticipation for future use without actually knowing what that future use will be. So biobanking itself is a huge undertaking that requires significant upfront investment in time, coordination, funding. And because these technological advancements can often place new requirements on the type of samples to be used in these technologies and how they're processed, biorepositories need to give careful consideration as to how we're collecting, processing, and storing these samples to ensure that our specimens can be used in the future. But you core labs are also important stakeholders in these conversations because if the appropriate specimens aren't available to your investigators to use, then your expensive new technologies are going to be vastly underutilized. And you don't want to spend a bunch of money buying a brand new piece of equipment only to find that the specimens you need either don't exist or are difficult to procure. So in my talk today, I want to go through a few biobanking considerations and why they matter, with a particular focus on human subjects. I'll go through a couple of lessons learned and real examples that I've encountered while interfacing between the research and biobanking worlds. And then lastly, I'll give some tips for proactive conversations that can be happening now between the research, the core, and the biobanking communities to ensure that when the time comes, there will be samples available to do the newest and coolest technologies. So some quick background on myself. I am a translational scientist and the manager for City of Hope's Legacy Program, which is a post-mortem tissue donation program, colloquially referred to as rapid or warm autopsy. And the purpose of programs like ours is to partner with our patients who wish to donate their bodies to science and to collect tissues that otherwise would not be available to us in the clinic while the patient is still alive. And one of my key roles is to understand how investigators intend to use these samples and in what applications so that I can ensure that our methods by which we collect and process these samples are compatible with the intended technologies. So based on my experience, having had many of these conversations with investigators, I would posit that for the most part, they are going to focus on the steps here on the screen, with the most thought being given to what kind of technology they want to use. And then next, they'll probably just default to whatever samples they happen to have on hand or are easy to get. And then even less thought is going to be given to the steps in the middle. And I'm sure that many core facility directors can appreciate that not all specimens are handled or processed in a way that's compatible for a given technology. And so in situations such as this, now we have to scramble to figure out how do we get these samples to fit with that technology. And I believe the reason why this happens is because many investigators don't understand exactly where their specimens come from and what's happened to them upstream. And in fact, quite a bit has happened upstream. There's the physical collection from the patient and the circumstances surrounding that collection. Was it a research-only collection, or is this specimen left over from a clinical procedure such as a surgery or a biopsy? How was it handled and macro-dissected? Once it was removed from the patient, where did it go? And how long did it sit there? Who touches it? And using what methods, what reagents? And finally, how is it stored? And all of this happens upstream and prior to distribution to the user, and all of which directly impact the quality and the versatility of the sample. So we all know that there are almost countless types of sequencing platforms available, and additionally, many of these platforms offer multiple chemistries depending on what exactly you're trying to look at. And these different platforms are going to be best compatible with different specimen types and less compatible with others. And while companies seem to be trying to make their platforms compatible with a variety of input samples, realistically, there's a specific sample type that's going to be best compatible for any given platform and chemistry that's going to result in the highest quality data. So I mentioned earlier that I've seen that many investigators plan the technologies first and worry about the specimens later, which introduces our biggest biobanking challenge. What should we even collect? So from a biobanking perspective, we really need to consider how we prioritize what types of specimens we're collecting and storing. Of course, the holy grail of tissue collection is always fresh tissue. You can do anything with fresh tissue, and an investigator who is handed fresh tissue can proceed to process in any way they wish. But if the sample is not going to be used right then something has to be done to preserve it, and each step of processing is, of course, going to limit the potential application of that sample. So there are pros and cons for the different types of samples that you can collect and store. FFPE is great for studying cell interactions, and you can do spatial transcriptomics, but it's not very good for doing DNA and RNA sequencing all the time if you're looking at traditional next-generation sequencing. Frozen samples are better for bulk DNA and RNA quality, but you're not going to be able to study unique populations of cells within your samples. Fresh specimens can be disassociated and cryopreserved into single-cell suspensions, where you can study single-cell sequencing, but you're not going, and you can identify unique populations of cells in these samples, but you're also going to miss out on how these samples interact with each other. So this is the challenge as we consider how to optimize our processes to meet the most of the research community now, while also anticipating the research needs in the future. So when we first began our program, our donation program, we were collecting both flash frozen and FFPE because we knew that we wanted to do bulk sequencing and histology. But initially, we didn't put a lot of collection or thought into our collection logistics. And so for the first few cases, what we ended up doing is we were collecting our FFPE and our flash frozen samples from different and distinct lesions, meaning one lesion was collected and frozen, and another lesion was collected and placed in formalin. And our collaborating lab was planning on doing RNA and whole exome sequencing from our flash frozen samples. But when they went to do the purification, the RNA and the DNA qualities were just absolutely abysmal, which was really strange to us because when we looked at our FFPE samples, they looked great. 90% tumor, very low necrosis. But when we went back to our collection notes and our photographs, we realized, our mistake. it had turned out that the sample we collected for the the sample we'd collected for our flash frozen was taken from a lesion that was particularly necrotic. And additionally, the sample that we used for FFPE was from a larger lesion, and it was very easy to cut it down to ensure that we only had tumor in our FFPE block. But the lesion that we used for our flash frozen was much smaller, and after we went back, we realized that we likely had some surrounding non-tumor tissue in that sample, which would have skewed the results of our sequencing anyways. So after some back and forth discussion with our lab, they decided to pivot to only using FFPE tissue, even though that this risked lower quality DNA and RNA extraction overall. We decided that at least we could validate the quality of the original specimen by H&E, and if we needed to, we could perform microdissection to ensure that we were only including tumor in our sequencing input. So this experience led us to modify our collection logistics, and in consideration of this conundrum, our program has developed a very specific cutting technique that allows us to collect both FFPE and frozen tissue from the same lesion, with the region intended for FFPE immediately adjacent to the flash frozen samples. So this allows us to not only validate our tissue quality, but it also gives us confidence when we're handing out these flash frozen samples 3 years down the line. Additionally, we decided that our samples that we were collecting for flash frozen would be microdissected to ensure that we are only including tumor tissue and no surrounding non-tumor. Our FFPE samples, on the other hand, can contain adjacent tissue, as these regions can be easily visualized in downstream experiments, and actually having a piece of normal tag can serve as a marker to indicate that this lesion did indeed come from a liver and not from a lung, and there wasn't some kind of labeling mix-up. And we can adapt this technique depending on the research needs. For example, if an investigator wants to collect fresh tissue as well, we can collect the FFPE from the middle to ensure that it best represents the biological characteristics of both the flash frozen and the FFPE samples. Another consideration we can consider is that depending on the intended research use, we can include additional surrounding tissue. And this might be useful when studying spatial transcriptomics or trying to understand the metastatic niche and tumor spread. Our second learning moment came from fluid processing. And similar to tissue, fresh is best, but you can't store fresh, so you have to process. So when we were trying to understand, how we should process our samples, we pooled our community to understand, what kinds of samples they're interested in, what applications. And we specifically asked, what is popular now, what's on the horizon? And overwhelmingly, we heard that ctDNA was hot. And unfortunately, that's about all we heard from them. But that was fine. So we asked what protocols do they use to process their specimens, and we began implementing those protocols in our processes. In particular, and this is important in a moment, processing for ctDNA involves a double high-speed centrifugation step. So everything was great, and everyone was happy for a while. And then cue the explosion of EV research, extracellular vesicles, which are now the newest and coolest thing to study. And that was great, we thought. We have so many samples that we've already collected, we're ready to hand them off to investigators. But it turns out, of course, that EVs are much heavier than cell-free DNAs. And the double high-speed spin that we had been using was causing our EVs to fall out of suspension. So lesson learned, now we process our plasmas at a much lower speeds, and for investigators that need those higher speeds, we make our protocols available to them, and they can process further if needed. Our last few learning moments came from interacting with the clinic. So many specimens in biobanks are actually leftover from clinical collections. In California, the law states that any clinical specimen removed from a patient in a clinical setting must first go to pathology before it's released for research use, which makes sense because patient care is always the priority. But it turns out that CSF collected in the clinic is often left at room temperature without EDTA for hours while the labs are being run. And we found out the hard way that unfortunately, by the time we get these samples back, any cells that had once been in there are long dead, and that these samples are not suitable for anyone trying to study circulating tumor cells. But on the other hand, EVs tend to be very stable, and these samples are appropriate for EV research. Conversely, if blood is stored at 4 degrees, which it often is, the CTCs are preserved, which is great, but the platelets begin to pump out their own EVs, which quickly overpower and outnumber any EVs of interest, making research very challenging. So in the context of clinical specimens, it's very important to understand these nuances up front so that investigators aren't wasting their money by attempting to use these specimens that are simply inappropriate or incompatible with their research and the technologies. So some key takeaways that you can share with your investigators. Please tell them that they shouldn't just plan the technologies, but they should also consider how their specimens are being collected from the very beginning. They should be aware that not all biobank specimens are going to be appropriate for their research and the technology that they want to use, and they shouldn't waste their time or their money performing assays on specimens that simply won't fit. You can encourage your investigators to have conversations with their biobanking leaders about the collection and processing methods that are being used, and encourage these biobanks to anticipate how emerging technologies might impact the technical requirements for those samples in the future. As for proactive conversations that can be happening amongst all of us, just poll your community to understand, not only what are the current research needs, but what's on the horizon, and how might that impact how samples need to be processed in the future. And before investing in new technologies, verify that there's an appropriate source of specimens to use, and if there's not, engage your biobanking leaders to brainstorm how you're going to address this new need for specimens. Biobanks should not put all of their eggs in one basket because technologies are going to continue to change and favor one specimen type over another, so it's best to process in multiple ways if possible. Choose processing methods that maximize potential applications and don't overprocess like we did. And lastly, there are always going to be situations where there is limited tissue quantity and you will have to prioritize one type of specimen over another. And in these situations, I would encourage you to think proactively about what technologies are the gold standards and what sample types are most appropriate for those and process accordingly. So that's all I have. Thank you very much for your time. I'm happy to take any questions if there's any.
It's on. Is it on?
There you
okay,
go.
There we go. A great talk. I have maybe a little bit question to the side. I just really need some opinion. If we have a longitudinal study that, let's say, we're going to do RNA-seq, and we're collecting samples over a year and a half, and we want to avoid, I don't know, minimum batch effect. Should we save them all, and then extract at the end? Should we extract them as we get in and store the RNA? Do you have a feeling?
That's a tough one.
Because if you extract all at the same time, then some of them are a year and a half old, some of them are a week old. And
Based on my own personal experience, I would extract up front and then save that extracted material until you have everything, and then do your libraries at the same time and run them all together.
Okay.
Because, the longer you wait for your FFPE, the more degraded it's going to become.
Great. Thank you so much.
Okay, I have a question. Okay, so is there any research approach to you with this great idea about the project, but they couldn't proceed it because of the lack of knowledge of this specimen?
Yeah, I mean, so when we work with our investigators, because we're specific to this, I am in the biobank, but more specifically, I work with, the tissue donation program. And so when we do work with our investigators, we really try to meet with them up front before we collect anything to understand what their research needs are, so we can collect it in a very specific way. Because like you saw in my presentation, if we don't know these things, then we can't really anticipate. In the situation with the EV research, we were so excited because we thought we have all of these samples we already collected for another project, and now we can maximize research potential. And then she dropped this bomb like, oh, by the way, you can't refrigerate any of these samples at all. And for us, it's had a big implication because sometimes, our donors are cooled down while we're waiting before the collection procedure, or if it's done at 2 a.m. and we can't get someone to come in and do the collection overnight, we'll put our blood at 4 degrees. And so it really changes the operations of how we're going to do that processing. So it's very important to know up front. Thank you.
Okay, next is Olivia from University of Michigan.
Okay. I'm going to apologize if I cough. I'm sorry. I'm still recovering from AGBT. But today I'm going to talk a little bit about how my facility kind of has to change on the fly in order to stay relevant and meet the research needs on campus. So this is our mission statement. It's very similar to most core facilities. Our goal is to stay as cutting edge as possible with the instrumentation and the technical expertise that's needed in order to support a wide range of offerings. And so a little background on my facility here. We have taken the approach that we are centralizing next generation sequencing, as short and long read, as well as all single cell processing and spatial omics assays on campus. So this is good and bad in a way. It's very easy on the customers to get work done. And so what we do is we have about 11,000 square feet. It's located on North Campus, if you've ever been to Ann Arbor. So we're a couple miles away from the main campus, but the ability to have that space that we need has been critical for our evolution over time, because we can expand as needed and bring on additional workflows. And so what this translates to for us is we delivered last year about 1 petabyte of raw data, and we service over 370 unique investigators annually. So with this sort of large facility, there's a complex organizational structure to keep it all moving. There's a lot of moving parts. So I have my, what we call our traditional sequencing team that's overseen by my assistant director. I also have a project manager that specializes in all the molecular aspects of the lab. And so all of our bulk frozen DNA and RNA comes through that facility. This is also the team that does all the work for anything downstream of the other workflows, right, that need library prep or sequencing. So we're really running sort of an assembly line type situation. I have a single cell and spatial team. They also have their pair, a lab manager that oversees the workflows and the staff, and then their project manager. These are very high-touch experiments, and so that requires their own dedicated team to lead. And then finally, I have what I call my dry lab. And that's a combination of my developers and my bioinformaticians. And from an evolution perspective and ability to adapt on the fly, my developers are key. In 2019, we started building our own LIMS, and it's still in production. And it is under construction at all times. And this is where my very talented developers are able to come in and switch up modules, add new content. And so for a centralized LIMS system that can take RNA or a tissue block and keep going, that's perfect for what we need to do. But this is where we're at. This is not where we started. So I took over the facility in December of 2018. At that point in time, we were not a genomics facility. We were called the DNA Sequencing Core, and we had a lot of legacy services, as shown on this slide here. And you can see some of these didn't really make a lot of sense to even still be in production. But they had done a couple of good things, right? They had started single-cell sequencing in 2017. They had their first Chromium device. They had come in and started to really see a nice interest there, and so that provided us a potential avenue for growth. They had a NovaSeq 6000. It wasn't in production, but they had bought it and procured it, and it was in the lab, and it was plugged in, but no one used it. So there were some things we needed to look at. And so one of the nice things, I guess, about moving to Michigan from Tennessee, because before there I was at Vanderbilt, is it's December. You don't go outside much. It's cold, it's snowy. It's a polar vortex, actually, that year. And so what I did was I stayed inside, and I talked to my staff. I talked to the customers, right? And I started kind of looking at the facility and formulating a plan for how we were going to proceed to make these changes that were going to need to happen in order to modernize the facility. So to do that, you need to have your community engagement, right? And so I was new, and we had a solid user base. We had a fantastic committee that had undergone this process of really looking at the facility and deciding what kind of director to take it to the next level. So we had that support, right? We knew we needed to make some changes. But to do that, you had to turn around and say, you can't do it all, right? There's no way you're going to please everyone, and you can't do everything. And so after accepting that, but you had to accept it, and you had to get your staff to accept it too. I sat down and we articulated a mission and vision statement. What did we want to be doing as we move forward, right? We can't do it all, and we had to pick a direction, right? And the direction my facility's at now is nowhere near where it was back in 2018. And from there, I actually, we're scientists, we like toys, we like to explore and discover, right? But we're also running businesses, right? And you have to kind of put on that business hat occasionally and say, this is what we're doing and this is why, and make those hard decisions. So what I did was I actually developed a business plan. And this was just bullet points of what I was going to do as I moved forward. What were the things I was going to consider as I made decisions on how the future of the core was going to look? And so a couple of things for me personally, I needed the core to be cost neutral. Not all core facilities operate that way. Not all core facilities really, thanks to institutional support, have to operate that way. My facility was not operating that way. I had a substantial deficit, and I knew that was going to stop. And that was going to make some hard choices on my part to kind of right that ship, but it was going to be done. So cost neutrality was key for me. Cutting edge. Legacy technologies are great, but you have to know when to let them go. And so making sure that we had that sort of kind of fundamental bullet point there that says we're going to always try to be a leader, not a follower, was key. And there's other things too. Institutional policies you want to make sure that you're looking at, right? Do you have the right staff? And staff are fantastic, but if you're going to go in a different direction, you might have to evaluate skills and see who can be retrained, or unfortunately, at times, when you might have to let someone go. And so these business plans are integral to my facility, and it's a little different than a lot of how facilities operate, but they seem to work for us, and we've been extremely successful in making a host of changes. So it's a risk anytime you do something, right? It's a science. We don't have an answer. We don't know what's going to work, right? And we want to stay cutting edge, but just as critical to being cutting edge is knowing when to let it go. Or maintain it, right? So I'm constantly looking at usage and demand, right? I'm looking for trends. Is there something in decline as something else is actually, gaining momentum? I'm looking at what the cost is to keep something operational. What are the benefits to the research enterprise? And also, again, does it align with our core mission? Is it fit within that rubric I established the first year I was here? And then I want to make sure and the technological relevance is there for my facility. Does it make sense? There are a lot of players and options in the field, and you have to be strategic in everything you do, because again, it goes back to that you can't do it all. And writing it down really does help. It's gonna help downstream, because these are, all your decisions from this point on are gonna have to be justified. You've got stakeholders that have to be engaged, right? And those stakeholders are your users, your faculty, your staff, right? Because all these changes are gonna impact them as well. So writing it down is really helpful. And there are always gonna be challenges, right? From a challenging, challenges beyond just budget and training, right? Funding is funding, and institutions choose to support core facilities differently, right? How you get instrumentation, how you pay for your compute, your data storage, right? All of those are going to vary by institution. Funding is obviously a challenge, right? And we all face that. There's also that staffing issue, right? I mentioned that earlier. Do we have what we need? Are they trained appropriately? Can we retrain? Or if we're going in a completely different direction, right? Do I have to hire a set of skills that just don't usually exist inside a genomics core? And that's what we did, actually, in one of our workflows. But more important, in my mind, not that those two aren't important, are the other things that you might not be considering routinely. And that comes down to infrastructure, right? Infrastructure can mean space. Do you have the space to shut something down and free up, a bench to bring something else online? Or if you bring on a whole new workflow, can you silo that somehow so that it's in its appropriate environment? As things become more advanced, everything is networked, right? Absolutely everything is networked, and those network speeds are going up, right? So are you going to have the right network capacity for whatever you're looking to do? Do you have beyond networks, cybersecurity is key, right? I work for a medical center. We take cybersecurity very seriously. Is what you're looking at, either shutting down or bringing online, fit within your institutional policies for what can and cannot be done? And sometimes that actually does dictate a choice in equipment, or the need to actually shut something down because it can't go on the network anymore, and the workarounds just don't seem feasible. Are you ensuring that you're not creating redundancy on campus, right? Resources are finite. We want to be fiscally responsible. We don't want to duplicate efforts. And so yes, I'm part of the School of Medicine, and we have this great little niche where we have fantastic support, but there are another 100 cores on facility beyond the 10 that are part of the School of Medicine, right? And so we want to make sure we're not duplicating effort, right? Because those institutional resources are finite. One of the other big challenges for us is resistance to change. I personally love change. That's why I'm in a genomics core, because tomorrow we're going to do something different than we did today, and that's fine by me. Researchers don't necessarily like that change all the time, and your staff probably hate it, right? And so there are things that we can do to engage, right? Because ultimately, the choices that you make going forward are going to impact what the researchers can do and what the staff are doing on a day-to-day basis, right? And so part of my plan is to try to engage them as quickly as possible. You're going to need their support, right? You're not going to do this in isolation. And so this comes back to having that business plan. You've kind of parsed down exactly what you need. And so for every single instrument or workflow that we're looking to shut down, maintain, or onboard, we do a separate business plan where we go through all of those rubrics and those bullet points and say, does this make sense, right? Does it meet our goals strategically? Can we cover all of our costs? I don't want to go into debt again, right? Is it going to be useful to the research community? We're running those metrics every single time that we're looking at shutting something down or turning something on. And keeping our users engaged and knowing what they need to know when they need to know it is key. And it's not just your users, it's your staff, right? And this is where having an implementation plan is extremely important. Knowing that you want to minimize disruption, and no matter what you do, you want to maintain your reputation, right? And not cause chaos on campus, because those are your stakeholders, right? Your researchers are the stakeholders, and you want to make sure that the changes you make are aligned with what their goals are. And finally, I don't care how much planning you do with genomics, you got to be flexible. This is a constant process. We are looking at this every single day. and trying to decide which directions to go to not only meet the current needs, but our future needs as well. And so a couple of ways we talk to our customers routinely are through our educational outreach seminars. We do introductory workshops. We're a very big facility. I have 27 staff. These are divided into 4 workflows. And so what we're doing in these workshops is, and I say we, it's not me, it's my managers that are hosting these workshops now, but they're educating our customers, right? We do these virtually. Our campus is huge. No one wants to jump between them for a meeting, but Zoom works fantastic for this. And we do 4 sessions a month. And what we're talking about in these sessions are, we're going through the options. What are each platform that we have, right, for each of these workflows? You know, if you're doing long read sequencing, do you want PacBio sequencing? Do you want ONT sequencing? And what are the pros and cons for the two, for example, right? So we're talking about choosing the right platform for their needs. We're going through our submission processes. I have a centralized LIMS system, so that means the interface that all of my customers have from my facility is the same. Input requirements are different, though. You know, the buttons you might have to press and the questions you have to answer vary. And so those are the kinds of things we cover in these workshops. We also look at cost. Genomics is not a one-line, one-charge assay. Everything we do is multi-pronged, and so we have an outward-facing cost estimator run by my developers in-house that allows people to guide them through an entire project. If you have a spatial project, do you need us to cut the blocks? I have an histologist, I can do it, right? So do you want us to cut the blocks? Do you want to section onto your slides yourself, right? And so there's all these questions we ask, and then we're able to give you a total estimate for the cost of your project. So we walk through that. We also go over some experimental design considerations in like the general sense. I say general sense because this is a group of people that all are going to have unique experiments at the end of the day, and we will do one-on-one experimental design meetings. But we ask that usually most customers that are new to our workflow or new to the core start here so they have a better sense of the offerings we have, and we can focus those conversations better. And then finally, we talk deliverables. We provide a lot of data to a lot of people routinely. And if they haven't worked with the Genomics Core in the past, we want to make sure that you understand what you're getting from us and how you're going to, if you're internal especially, leverage the university's infrastructure in order to make sure your data can be transferred to you for your ownership and then safely backed up. So this is all done in our workshop series. Not only is this a great way for my managers to work directly with the customers and gather feedback on what their needs are, currently, but ideally, they're gonna want in the future, they're working together because we're not doing these one-on-one conversations where I have the same meeting 12 times a week, where I explain the 16 options for spatial transcriptomics now, right? They're actually collaborating together and they're feeding off each other. So it's a great way to get feedback from our customers. Vendors are key too. Our vendor partnerships are extremely important to us. I personally can't meet with all of them all the time. This is where I empower my staff. So we do one-on-one meetings monthly or quarterly with a lot of our key players. But our staff is fantastic. They're able to engage with these vendors as well, bring information back to me, kind of keep their ear to the ground so we know what's coming. It's also great from their career development opportunity, right, to actually engage with some of our vendors. Seminars are important for us. We have at least an event a week, a month, sometimes 2. Every month except August. Ann Arbor empties in August. We never have anyone there, so we don't do anything in August. Other than that, there's always 1 or 2 events a month, and sometimes these events are to look at building a business for something we already have established. Sometimes the event is for looking at something new that we're thinking about bringing online. Sometimes it's a user group meeting where we're just actually trying to bring our customers together in a more collaborative environment. And sometimes it's a focus group. If we're looking at having to sunset something, we need that communication, right? And so town halls or focus groups are key. So there's always something going on inside the core to kind of help guide our path forward. We leverage core grants. They've been pretty successful at my institution, turning pilot projects into really large experiments. And then something we've done over the last couple of years that has taken off is an annual holiday open house. It's an all-day event. We have vendors, we have talks, we throw open the doors of the lab, we do tours. I actually talk very little this day. My staff are the ones talking to the customers. They're walking the customers through how it looks to put a slide on a site assist, for example, or what the robot is really doing to their samples as we make libraries, right? They're the ones engaging. This gives them that opportunity to talk to the customers. It's also a great opportunity for our vendors. We have vendor tables, we have lightning talks, we have hot cocoa and holiday treats, and like I said, it's an all-day event. It grows every year, and it is extremely popular. And again, it's fostering that engagement, so you know kind of which direction you're going to need to keep going in. You've got to stay connected. And I'm not going to spend a lot of time on this slide, it's very busy, but this concept of having that business plan, we went out and said we want to do spatial transcriptomics, right, in 2019. It wasn't a thing then. And so this is how having that business plan and deciding at each step, if we're going to onboard something new, bring on new skill sets, it was fast. And now we're certified service providers for multiple platforms. We have multiple instruments for certain platforms. It's a beast, and it continues to grow. And I would say it's been a successful way of bringing on new technology just because we haven't lost any bulk sequencing on campus. We're still at a consistent 5-6% growth year over year. 75% of my recharge revenue now is single cell or spatial because we're able to adopt something quickly and bring it online and meet those researchers' needs effectively. So hopefully I've convinced you that you want to stay as cutting edge as possible. And the way that my facility has opted to do this is really kind of treat it like a business, where we are using our mission and vision statement and our business plan to make strategic decisions, and that you absolutely have to have engagement of your stakeholders in order for anything to be a success. And that is it. I know I have 27 people. Not a single one of them likes to have their picture taken, so they provide pet pictures instead. So with that, I'm going to stop, and I'll be happy to take any questions.
Hi, I'm Barbara from Stony Brook University. Great talk. Thank you very much. I'm actually being promoted. I need to set up the Genomics Core at my university, so that was very useful. I might hit on you later. So my question was, it's hard to let things go. Very hard, especially if you're a perfectionist and an overachiever, but needs to be done, as you point out. It's part of good planning. And so I was wondering, for the things that you cut that were not cost effective, what do you do? Do you outsource? You help the researchers finding another way to get it done?
How we go about it depends on what we're talking about, right? In some cases, something gets shut down because honestly, it's lack of use. And we're able to transition the 1 or 2 customers to something else internally that meets their needs. In other cases, like when we just chose to outsource Sanger, we formed a task force. We had a task force that comprised of people in our community, but also my staff itself. And I tasked them to say, okay, so what do we do? Do we need to upgrade our instruments? Do we need to automate more? Do we outsource it, right? I was not part of that committee. I was part of making the final decision, but we engaged the staff to make that process happen, right? And then it has to be strategic, you have finite resources. Did it make sense to be cutting edge and just do Sanger sequencing, right? That was gonna limit my capital funds for other things, right? And so it's not an easy process, but you have to make those tough decisions. And unfortunately, with something as large as Sanger, it's not only getting the community involved and making them understand why we're doing what we're doing. We had to RIF people. I had to RIF 6 people when I outsourced Sanger, right? Because the skill sets that were running Sanger were not the skill sets I was going to need to build spatial, right? I had to hire a histologist, for example, right? So it's not easy, but it is doable. Again, you have to kind of disconnect from the emotion a little bit and say, what's the research really need?
Thank you. And one last question is, how do you make the decision to go back if things don't work as planned out? And how do you reassess? How often, how quickly, promptly do you reassess?
Literally on the fly sometimes. Sometimes you buy an instrument and it doesn't work as planned, right? And it maybe will over time, but hopefully you haven't shut down the last one that was working, right? And you have to pivot. You know, I had to pivot from a NovaSeq X Plus back to a 6000 for a while because the data was subpar for certain libraries, right? So you have that flexibility is really important. So you don't want to, for example, when we outsourced Sanger, we didn't have a hard stop, right? We had a transition period. That comes where that plan is important, right? So that you can pivot, you know, until everyone's comfortable and move forward.
Thank you.
Hi, thank you very much. I'm also Barbara. I'm from Arizona. I have a couple questions about your LIMS. Now, I understand you have a large computational and programming group, and that certainly helps with a homebrewed LIMS. But my questions are, one, is it used by both your lab for tracking on the bench and order intake? I think I heard you say that. And then also, if it's used for order intake, do you have both internal and external customers placing orders there? And then the third question is it used by any sister facilities at your institution?
All very good questions. So the front end for user interface for my LIMS system is iLab.
Gotcha.
Okay, so customers start with iLab, and we use the API to pull everything into our LIMS system, and then we have a lot of automatic messaging and pricing that gets pushed back, right? So we're using iLab. It's not our favorite, but that is our intake, and it's the same for internal or external, right? Currently, my LIMS is only in my facility. We are in the process of deploying it to another facility. We've just started that. It's difficult to get to the point where there's enough bandwidth because every time we turn around, somebody drops something new, and my developers get tied up in you know, modulating the LIMS again. But that is something that we can do. Because of the variety of what we see, it is going to be something that other facilities are going to be able to use.
Thank you very much. That's very cool. And kudos to your developers who got that all under control, because that's a big ask.
Yes, it is. My developers have a really good understanding of basic science, which I don't expect them to have, but they sit with protocols and my managers and technicians, and they figure it all out. They're fantastic.
Hello, not Barbara. Okay, my question was about, because I'm at this stage where, you know, the engineers are coming in, do we need gas? Do we need this, right? We're at that stage. And I was trying to think, what's going to be next after spatial? And I love that in 2019 you were thinking about spatialomics, and now it's 2025. What do you think is coming down the pike?
Oh, that's hard. Anything and everything. I've learned not to try to predict. I get a crystal ball out a couple times a year and I gaze into it. What we're seeing is a lot of multi-omics, right? And so one of the things that we're doing a lot of and seeing a lot of interest in right now inside my genomics facility is proteomics assays. Proteomics assays for bulk perspective, Olink, for example, or for spatial, or for single cell, right? And so I think the layering on is going to be probably the focus for the next year or so. And what we're doing is just how to gather more information from a single specimen. Okay.
Thank you, Olivia. All right. Hey, I'm Minhee. I'm learning the Genomics Core at Cleveland Clinic. Yeah, even though our core is way smaller than yours, but still the users, our core is learning kind of same thing over and over, even though like less requests there. So it's all similar workflow, I guess. Researcher reach out to you, consultation, I have this sample, I want to do this, can you consultate me like a single-cell sequencing or concurrent sequencing, and then we just figured out everything, and then we got the sample, and then do the sample QC, library prep, and sequencing, and check all those data, and then send them to user, right? So this actually the consultation takes way more than actually doing the QC and library prep sequencing, because we have like real like a good workflow and a good technician together. But like one time actually, recently, this researcher, she's an MD, and then she said, oh, I have a lot of great sample in the biorepository, and then I know there is single-cell sequencing, amazing technology, and I want to do it, and I have like 50-something samples. Can we do this? So, oh, absolutely, let's do this. And then I talked to the biorepository person, and then she said, we only have the frozen pellet of cells. Can you do single-cell sequencing? We can. So we have to consultation again, like, oh, no, how can I? So yeah, like, it's really surprising when you talk with people, like, they have really great imagination or, like, plan, but, like, the reality, like, it's very lack of reality. So we have to go through, there is mRNA or, like, total RNA sequencing. What's the difference? And then I have to explain this. And then, okay, I have 7 control and then 52, like, diseases, but there is a lot of different diseases group. But you have to think about how many samples per group also to compare the sequencing data, right? So all those things, you have to do this. So how many of you have the sequencer in your core? Oh, that's great, a lot. Okay, how many of you have like really amazing, like NovaSeq XP, X Plus? Oh, so rich. Oh yeah, I forgot to ask you, how many sequences you have?
9.
9, oh my gosh. So actually, I forgot to ask you again. So, you know, the genomics field evolving so fast, there's a lot of technology, but we cannot actually upgrade the really expensive sequencer every time it's available. So, Olivia, you have luxury to do that?
I have to make a business plan for it.
Oh, wow, so jealous. Okay. So, but luckily we have NovaSeq 6000, even though we have very small facility, but yeah, it's really great to have it. Other than that, we have to send out all those samples to outside, right? So we are in kind of dilemma right now. Should we collaborate with outsourcing to send out all this sequencing? Because like NovaSeq XP is way cheaper than NovaSeq 6000, right? So, and then when the small labs comes with a very small budget, but their ambition is not that small, right? So I'm just trying to work with them how we can get all those data, what they want. So this consultation takes a lot of time to do it. And then also there is like a single cell sequencing, like bulk RNA sequencing, and then like you have to bring this RNA in the dry ice. You have to tell them sometimes, because sometimes they bring this RNA in the like zip bag, and then like, oh, here's your my RNA samples. Ooh, please don't do that. So sometimes you have to really talk about very basic thing, how to prepare your sample, how you deliver the sample, right? And then 10x, of course, you guys all know 10x Genomics, and then they develop a lot of newer technologies, and then now they have these multiple samples with its 1-gem generation using the fixed cells, and also a lot of different kind of library kits. So it's really hard to keep up like all those different kind of methodology, and also like always user ask you like how much per sample. But like Olivia said, it's not that simple, right? So sometimes you have to go back to quality control everything, and then like, yeah, how many sequencing depths you want, you know, all those things you have to consider. So this consultation takes a lot of time. I mean, luckily Olivia has a lot of different managers, but I have to do all those things, making a call, consultation, everything. So I want to just point out one thing, like even the single-cell sequencing, usually it's poly-A-tail-based capturing, right? But now this with the fixed RNA, fixed cell, it's probe-based capturing for the single-cell sequencing for 10x. So it's kind of different. So when people come with you this very precious sample, patient sample with like a CAR-T treatment, but when you do just probe-based capturing, you only capturing the genes you already know about it, but you don't know what different kind of maybe they do want some kind of newer marker or something, then you have to go back this poly-A tailing capturing, but it's more expensive. But again, people think about price, so there's a lot of things you have to consider. Yeah, and then also for the bulk RNA-seq, there is mRNA sequencing, total RNA sequencing. You still have to explain them. And then there is low input mRNA, the quality and quantity, especially from the FFP RNA, it's very degraded. So some kit, like Takara one, like a Pico input kit, is really good with this FFP sample, low quality quantity samples. But there is also different, like when you do rRNA depletion. So Illumina usually they RNA depletion in RNA level, but Takara, they make to cDNA and then deplete all those rRNA thing. So sometimes we see a lot of like rRNA content in the sequencing data. That means that some of the method is not very efficient to remove those rRNA. Then like you targeting like 20 million reading per sample, and then 10 of them goes to the RNA. You cannot use them at all, and that's a lot of wasting money. And then so I ask them, so it's really important you communicate with vendor whenever you have question. So they are expert. I ask them that sometimes that method doesn't actually remove any mitochondrial RNA. So there is a lot think about all those things. So if you're aware of it before you consultation with your user, and then you kind of expect what kind of data you can get, even though we don't actually analyze data at all, but I'm willing to learn all those beforehand. And then like we learned ChIP-seq today. This is really amazing methodology when we want to do this ChIP-seq. It's really great. So also, it's very important to communicate with your staff and then training your staff. So once we got new technician, we try to make her or him master one library prep very well. Because library prep itself, like there is a lot of different kind of library prep protocol, but once you master one thing, it's easy to adapt on a lot of other things, something like that. So it's really important to like communicate and then training your staff. Actually, we do that every Monday morning, like a lab meeting, even though we are just core, we don't need to present anything, but it's really good to keep communicating together what kind of project we have last week, how it's going, and then next week, Sometimes we just got the samples just today or tomorrow. We cannot like really plan ahead, but still it's really important. I care about you and you care about me. We are team, the communication together. So we don't point a finger or something fail. So we always talk each other. Like if there is some weird things going on, we just present together why this is it. We should reach out to vendor, that kind of things we do. Oh, so the sample QC. So we already talk about all these important things, what's the sample you have to bring us, but still you see a lot of like different quality of that. So when you do the tape station, like here, like rinse score, 10 is perfect, but a lot of degraded sample over there. Sometimes we got like really unique kind of things, so then we reach out to company and then ask about their insight about that. And then also sometimes we have DNA contamination in there, so we have to ask them, did you treat the DNA or not? If not, willing to do it. Some people, I don't know, they don't want to do it, just let's do it. But like, can you actually trust the data later? And then single cell sequencing, it's also really important how to prepare your samples, because if it's not intact cell, intact nuclei, there's a lot of ambient, like free-floating mRNA in your samples, right? And then once you sequence them, how you know it's that gene expression from your cell or just background? It's so hard to tell, so it's really important to have this higher viability, more than 70% viability, have this single cell sequencing. That's really great to have. And then even fix the cells before they start to do the fixing cells, that's always they have to consider to do it. So now, quality control is done. So going to the library preparation. So this time it's going easy because our staff is well trained, they know what to do, even though we have very low users, like input something, but some people want to do very unique library prep. But like I said, one person trained with really good, like one library prep, they can just look at another unique library prep protocol, and then they just tackle this. So after that, we check the library peak with the tape station again, or bioanalyzer. Sometimes you get the weird peak again, out of this library site, and then we reach out to the company and then discuss about this, and then always update users through the process. And then we use QuantaBio qPCR1 before, after making the pool for the people sequencing. So some qPCR, the reagent is kind of different than others. So whenever you change the reagent kit, it's better to check ahead before actually totally change the reagent kit. Of course, there's a lot of sequencing, and we have Nova 66000, MySeq. There is like great Element Bio one, and Nanopore. So it depends on you want to do the short reading or long reading. That depends on what user really want. And then you really have to plan before actually the sequencing for sure during the consultation. And once it's done, so now there is a 10x flex. There's a lot of samples all together, but there's only one index. So we cannot demultiplexing after sequencing. So you have to go to the cell ranger, 10x cell ranger, and then you have to do it. And then you see all this, how many cells actually captured by sample, all those things. It's kind of newer. We are just learning all those things. And then you have to make sure this sequencing data is great, the QC checking everything, and then all the reads are the user desired read, we reach that. If it's not, we do the resequencing for them for free. It's expensive, but we have to do that. It's really hard to equally pull all those samples. There's like 10 samples, 20 samples, 30 samples. You do the individually tape station, qubit, and then you pull them, right? But I don't know any better way to pull. If you have any idea, like really perfect to quantify all this library, pull them together, so please let me know. Other than you just do the qPCR individual sample, it's really hard to do it. But yeah, the best way we do this qubit and tape station together. So when we deliver the data to internal users, we have the shared folder under our the Cleveland Clinic. So that's easy to transit right now. And then sometimes we have the external user, and then we use the FileZilla for the transfer. You guys use that kind of thing? No? Oh, okay, some different method you guys can use. So, for now, there is no limitation. We can store the sequencing data, but I don't know, in the future, maybe there's a limitation, so that also we're gonna figure out later, something like that. So we are playing really critical role to support all these amazing researchers, small or bigger, something like that. So I really think it's really important to educate people, the users, about this technology, and then always, do the right control, the quality control for the sample and sequencing data, all those things. So I think this, it's the right on time. So thank you for listening, and if you have any question, I can take the questions. Thank you. Okay. Oh, you have a question? Oh, you have a question? Oh, okay.
Just out of curiosity, Olivia said she's 75% of her customers have single cell or spatial transcriptomics. I was wondering, what about you?
Yeah. So it's kind of interesting. Every year kind of different. So like 2 years, it was like about 50%, something like that. And then last year, about like 10%, 20%. And then the thing is, like, some of people actually send the sample to University of Chicago from our facility because it's cheaper because they have really amazing everything, right? So sometimes like that, but it's kind of vary every year.
And I have one more question. Sounds like you guys are offering tremendous support with all kind of library preps that people come to you and you say, okay, give me the manual, I'll do it.
Yeah.
What are the challenges that you find in doing that?
Yeah. So, I mean, like, if it's really different, then we have to say no. But if it's a little bit of, like, differences in there, and then if they actually give us this library prep kit, because usually it's like a 24 sample or 96 sample, but we cannot have that for unique library prep, right? So we have to ask users, can you provide us all the reagent with the protocol? Then we just look through it, and then if we have all this right instrument, everything, then we said yes. And then we just work through it, just to study ourselves, and then contact to the vendor, if we have any questions. So, I mean, if you really busy, you cannot do it, but if you have time, we just try to do that.
And one last question. How are you keeping the reagents in stock, at least for the most used kits?
Yes, yes.
And
like
the other
RNA, because mRNA, total RNA, small RNA, yes.
And the weird ones, you ask the customers to purchase them themselves, is
yes,
that right?
and then send them back.
Okay.
Yeah.
Thank you.
Yeah, no problem.
Hi. I have a general question for the panel.
Oh, sure.
Just in terms of, like, in the spirit of adapting quickly, I guess. Like, there's a lot of vendors, a lot of techs, a lot of kits, and you want to adopt it early, so it's useful, but you don't want to adopt it too early before it's proven. How do you walk the line, or like what are some considerations to think about in terms of, should I test this new kit that's out in the market by vendor X? Do you constantly budget in like R&D like allocations where you're like, okay, there's a new kit, theoretically could be cheaper, but is the quality there? Do you take the vendor's words for it? Do you test it out in-house? I'm just curious, how do you think about when you're ready to like give it a shot? How do you make that call?
Yeah. It's a small facility here, core. Actually, we actually tackle the everyday task, right? So we don't have time actually sitting there reading all the news, something like that. But it's really important to contact with this vendor all the time, and then they actually inform us sometimes. So, and then you can see the emails sometimes. So it's just to try to read all those things, try to catch up those things. But even though it's really expensive, we cannot tackle, we have to purchase like a real big equipment or something, but I'm trying to like have the knowledge about it so I can reach out to vendor or even other institute to help these users to support their the research, right? Even though we cannot do it. So that's how kind of adapt this newer technology. How about you?
So for us, it depends on what we're talking about, right? If it's a library prep kit, periodically we do bake-offs, right? And that's stuff that's inside my budget to do. And as for your first part of your question, sometimes you have to take a risk. I had the 10th Xenium, is that right, Trish? The 10th Xenium installed, and it had a plethora of issues when I first brought them online. But it's one of our biggest workflows, and we now have 2, right? And so sometimes you have to take that leap of faith, and it has to do with, as I'm going through and looking at competing technologies, because you can't do them all at the same time, and you won't be able to test every single one of them, you have to rely on some of your relationships, right? What's your support like for a certain vendor, right? What is the infrastructure requirements? Something that's like a non-starter for me at the University of Michigan, actually, because I'm part of the hospital, I can't do cloud-based analysis right now. So anything that's cloud-based kind of put something like that to the bottom of my list, right? Because I just couldn't get it through. information assurance, right? So, but at the same time, you have to take that risk if you want to be a leader and not a follower. So I understand wanting to be cautious, but this is, it's Vegas, it's a gamble, and you want to take those risks, right? So that you aren't behind at the end of the day.
Yeah, and so you're always, is it fair to say, like, good relationships with the vendor, but you would still test it for yourself to make sure you can convince yourself that it's good?
Sometimes, yes. If it's a kit, absolutely, we'll test it ourselves. But sometimes you have to go on blind faith, and it's based on the history you have with usually the company, honestly.
Yeah, sometimes company give us free kits, right, to test.
Sometimes if you have a really good investigator who'll be like, yeah, I can throw you a couple of samples to give it a shot, that goes a long way because you can't always get real world samples. You can't just buy PBMCs or something, or you don't want to use cell culture, right? We do skin first on everything because we have this investigator who's always like, yeah, go ahead, try out some skin. You know, if the data looks great, maybe I will switch. And so fostering those customer relationships is also really important.
Yes. That's my single cell spatial lab manager, so.
Awesome, thank you.
Hi, my name is Brian from ARC Institute. I have a question maybe also for the panel. How do you manage, you all have like extensive user consultation meetings. How do you manage expectation in terms of like turnaround, for example? Because I know everybody wants their data yesterday, so.
You want me to do that?
How you do that?
Okay. Okay, so managing turnaround, be as transparent as possible. Almost all of our workflows, with the exception of spatial, are 4 weeks or less, start to finish. I say spatial because we are limited with certain platforms because of scan time, for example, and the backlog we might have. So set those expectations early so that they're not surprised. And then what we found we need to do sometimes is prioritize and really talk to customers at the front end if they have, and again, this is mostly spatial for us, the in situ platforms that are going to run as long as they have to run and there's nothing we can do about it, is we have to balance, long-term projects with other users, right? And it's just setting those expectations. It's one of the reasons why I have a project manager for both our molecular workflows and our single-cell spatial. So there's a point of contact so those conversations can happen. And we are large. A lot of our users are actually large as well, and they have their own project managers too. And so my project managers coordinate with their project managers, right? And it's basically just to be as transparent as possible. And again, we just do the best we can. And sometimes there are issues. An instrument goes down, and we have to just keep them informed. But for the most part, it's 4 weeks for everything except spatial. Spatial is 6 weeks or less from image approval. There's a lot of QC that goes into spatial, though, that delays things.
Yeah, like I said, we also, when we do the consultation, I just give them all the honesty about what to expect about this, the quality of the sample or everything. So once we check the quality of the sample at first, we just share all the QC data, especially if there is something goes wrong, and then we just talk about, you still want to proceed with this, process, even though it's not guaranteed. We cannot guarantee the good quality of data later. So we just being really honest, and then we share every conversation with the vendor, all those data. So we haven't had any issues because, all those things. So, all right, thanks for coming, and then have a great evening, guys.